Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 7.5 Hardy-Weinberg equilibrium. So what will we be learning today? For today's lesson, we're going to look at what is the Hardy-Weinberg model, what are the conditions under which allele and genotype frequencies will change in populations, how are allele and genotype frequencies calculated using the Hardy-Weinberg equations, what do changes in allele frequencies provide evidence for? And then finally, what are the impacts on the population if any of the conditions of Hardy-Weinberg are not met? So Hardy-Weinberg is a model for describing and predicting allele frequencies in a non-evolving population. A population in Hardy-Weinberg equilibrium is not evolving. Frequencies of alleles and genotypes stay the same generation after generation for a population in Hardy-Weinberg equilibrium. 5 conditions that must be met for a population to be in Hardy-Weinberg equilibrium include: 1, a large population, meaning no genetic drift. 2, an absence of migration, so no gene flow. 3, no net mutations, so no genes are modified, deleted, or duplicated. 4, random mating, so no sexual selection. And then finally, an absence of selection, so no natural selection. These conditions are rarely met, but they provide a valuable null hypothesis. Allele frequencies in a population can be calculated from genotype frequencies using the Hardy-Weinberg equations. P squared plus 2pq plus q squared equals 1 is one of those equations, where P squared is equal to the frequency of the homozygous dominant genotype, 2pq is equal to the frequency of the heterozygous genotype, and q squared is equal to the frequency of the homozygous recessive genotype. Remember, this is used to determine genotype and or phenotype frequencies of individuals in a population. P plus q equals 1 is where P is equal to the frequency of the dominant allele, and q is equal to the frequency of the recessive allele. This is used to determine the frequency of a particular allele in a population. Now, changes in allele frequencies provide evidence for the occurrence of evolution in a population. These factors can disrupt Hardy-Weinberg equilibrium and change allele frequencies within a population. One of these factors, mutations. Changes in genes can occur through random events that delete, insert, or substitute nucleotides. Another factor, non-random mating. Individuals choose to mate with another based on certain traits. Gene flow is another factor where new genes can be introduced to populations, so migration of individuals in and or out of a population. Genetic drift is where changes in the allele frequency within a population can occur due to random environmental events. Think bottleneck founder effects. And finally, natural selection. Alleles improve or reduce fitness for individuals to survive and reproduce in a given environment. So let's look at an example. We're going to look at a population of beetles in Hardy-Weinberg equilibrium. The population consists of black and red beetles. The dominant allele is black, and we'll use capital A for that allele. And the recessive allele is red, and we'll use lowercase a. The black beetle population consists of the homozygous dominant and heterozygous genotypes, and the red beetle population consists of the homozygous recessive genotypes. For this population, the dominant allele, P, is equal to 0.3, and the recessive allele, Q, is equal to 0.7. With P plus Q equals 1, so 0.3 plus 0.7 is equal to 1. Now we're going to look at what is the frequency of the homozygous dominant genotype. Well, we're going to use the equation P squared plus 2pq plus Q squared equals 1. P squared is the frequency of the homozygous dominant genotype. So P squared is equal to 0.3 squared equaling 0.09 or 9%. For the heterozygous genotype, it is equal to 2pq. which is equal to 2 times 0.3 times 0.7, which equals 0.42 or 42%. And then finally, what is the homozygous recessive genotype? Well, that is equal to Q squared, which is 0.7 squared equaling 0.49 or 49%. It is important to be mindful of your decimals when you're working with the Hardy-Weinberg equation. So now let's look at an example that you're going to work through. For this problem, I would like you to pause your video, solve the problem, and then push play when you're ready with your answers. Welcome back. For this question, you were looking at what is the frequency of each genotype in this population and what is the frequency of the dominant phenotype. The frequency of the homozygous dominant genotype is 0.36. The frequency of the heterozygous is 0.48. And the frequency of the homozygous recessive is 0.16. Now, what is the frequency of the dominant phenotype? Well, that is going to be equal to both p squared and 2p cubed because both of them are going to exhibit the dominant phenotype. And the answer there would have been 0.84. So now we're going to practice some more. For this practice, you're going to perform a mathematical calculation, including mathematical equations in the curriculum. So for this practice problem, I would like you to hit pause, read the question, formulate your answer, and then hit play again. Welcome back. For this question, you were supposed to look at, based on the information provided, which of the following best represents the calculated differences between the isolated population and the general population. Your answer should have been D, that the frequency of the Ellis-Van Creveld allele is 0.0447 in the isolated population and 0.0026 in the general population, which suggests that genetic drift has occurred in the isolated population. A couple key points on solving this question is that Q squared was given to you, where in the general population, it was 1 in 150,000 live births. And then in the isolated population, it was 1 in 500. And the genetic drift occurred due to those isolated populations. So what should you take away from this lesson today? First, Hardy-Weinberg is a model for describing and predicting allele frequencies in a non-evolving population. 2, the conditions for a population to be in Hardy-Weinberg equilibrium are a large population size, absence of migration, no net mutations, random mating, and absence of selection. The allele and genotype frequencies can be calculated using the Hardy-Weinberg equation where P and Q represents alleles, and P squared, 2PQ, and Q squared represent the genotypes. Changes in allele frequencies provide evidence for the occurrence of evolution in a population. And then finally, a population can evolve if the conditions of Hardy-Weinberg are not met with small populations, being more susceptible to random environmental impacts than large populations. Well, I hope this video has been helpful. Until next time, thank you.